Welcome to the Bio Whisperer channel. Our topic today is on a perspective overview of the Omics sciences. Since the process of mapping and sequencing the human genome began, new technologies have made it possible to obtain a huge number of molecular measurements within a tissue or cell. These technologies can be applied to a biological system of interest to obtain a snapshot of the underlying biology at a resolution that has never before been possible. Broadly speaking, the scientific fields associated with measuring such biological molecules in a high-throughput way are called omics. Examples include proteomics, transcriptomics, genomics, metabolomics, lipidomics, and epigenomics, which correspond to global analyses of proteins, RNA, genes, metabolites, lipids, and methylated DNA or modified histone proteins in chromosomes, respectively. There are many motivations for conducting omics research. One common reason is to obtain a comprehensive understanding of the biological system under study. Genomics is the new science that deals with the discovery and noting of all the sequences in the entire genome of a particular organism. The genome can be defined as the complete set of genes inside a cell. Genomics is, therefore, the study of the genetic makeup of organisms. Determining the genomic sequence, however, is only the beginning of genomics. Once this is done, the genomic sequence is used to study the function of the numerous genes, functional genomics, to compare the genes in one organism with those of another, comparative genomics, or to generate the 3D structure of one or more proteins from each protein family, thus offering clues to their function, structural genomics. Genomics is an entry point for looking at the other omics sciences. The information in the genes of an organism, its genotype, is largely responsible for the final physical makeup of the organism, referred to as the phenotype. However, the environment also has some influence on the phenotype. I would thank you for helping to share your love for this science channel by clicking the like button and subscribe for future updates. Myself and my team of fellow retired educators, Dr. Biotech Whisperer's team of 4 curators including Dr. Owl, will love to hear your comments and suggestions for future topics. With that out of the way, let's continue the discussion. Proteins are responsible for an endless number of tasks within the cell. The complete set of proteins in a cell can be referred to as its proteome, and the study of protein structure and function, and what every protein in the cell is doing is known as proteomics. The proteome is highly dynamic, and it changes from time to time in response to different environmental stimuli. The goal of proteomics is to understand how the structure and function of proteins allow them to do what they do, what they interact with, and how they contribute to life processes. An application of proteomics is known as protein expression profiling, where proteins are identified at a certain time in an organism as a result of the expression to a stimulus. Proteomics can also be used to develop a protein network map where interaction among proteins can be determined for a particular living system. Proteomics can also be applied to map protein modification to determine the difference between a wild type and a genetically modified organism. It is also used to study protein-protein interactions involved in plant defense reactions. Metabolomics. Metabolomics is one of the newest omics sciences. The metabolome refers to the complete set of low molecular weight compounds in a sample. These compounds are the substrates and byproducts of enzymatic reactions, and have a direct effect on the phenotype of the cell. Thus, metabolomics aims at determining a sample's profile of these compounds at a specified time under specific environmental conditions. Metabolomics can be used to determine differences between the levels of thousands of molecules between a healthy and diseased plant. The technology can also be used to determine the nutritional difference between traditional and genetically modified crops, and in identifying plant defense metabolites. Integrating what we know so far, genomics provides an overview of the complete set of genetic instructions provided by the DNA, while transcriptomics investigates gene expression patterns. Proteomics studies dynamic protein products and their interactions, while metabolomics is also an intermediate step in understanding organisms' entire metabolism. Transcriptomics. Decoding the genomic DNA is one step towards understanding the process of expression and functional genomics. The basic flow of genetic information in a cell is as follows. The DNA is transcribed or copied into a form known as ribonucleic acid or RNA. The complete set is subject to some editing which occurs in the nucleus to become messenger RNA, which carries information to the ribosome, the protein factory of the cell, which then translates the message into protein. The transcriptome is the complete set of RNA transcripts from DNA in a cell or tissue. The transcriptome includes ribosomal RNA, messenger RNA, transfer RNA, microRNA, and other non-coding RNA. In humans, only around 2% of the genome is represented in the transcriptome as protein-coding genes. The two dominant classes of measurement technologies for the transcriptome are microarrays and RNA sequencing, also known as RNA-seq. One of the older approaches is using microarrays, which are based on oligonucleotide probes that hybridize to specific RNA transcripts. It is being replaced with the more recent and modern approach RNA-seq, with key advantage in permitting for direct sequencing of RNAs without the need for probes. Some of the platforms include Chorus CAD, Oncotype DX, MamaPrint, and AlloMap, amongst others. Next we will touch on the epigenome. The epigenome broadly consists of reversible chemical modifications to the DNA, or to the histones that bind DNA, and produce changes in the expression of genes without altering their base sequence. The epigenome can vary substantially among different cell types within the same organism, where epigenomic modifications can occur in a tissue-specific manner, in response to environmental factors, or in the development of disease states. Such epigenomic modifications can persist across generations. From a biochemical point of view, epigenetic changes that are measured at high throughput belong to two categories. Firstly, we have the methylation of DNA cytosine residues, at CpG, as well as secondly having multiple kinds of modifications of specific histone proteins in the chromosomes, also known as histone marks. Another upcoming mechanism after DNA editing by CRISPR is the field of research on RNA editing for epigenetic changes in gene expression, which is being measured primarily by transcriptomic methods. The Omics sciences does offer potential benefits, and these are summarized below. Firstly, we see technological improvement and innovation that will progressively offer new opportunities of innovation for companies and entities in different fields. In terms of transversality, the extended application of the Omic technologies could be applied in a huge variety of sectors and applications, including in health and medicine. In terms of healthcare and medical application, Omic sciences are gradually integrating personalization, where advances in our understanding of cellular, molecular, and disease mechanisms allow patients to obtain more individualized treatments, especially in the field of personalized nutrition. In areas of new product and therapeutic development, we see value in creating new products with more health impacts that satisfy social trends and consumer needs. As research progresses, our expanded knowledge and understanding from the findings of Omic sciences will continue to deepen our awareness of the system biology and the mechanism of action. Genome-wide association studies have identified thousands of genetic loci associated with complex traits, including a wide variety of diseases. Despite the successful identification of associated loci, interpreting GWAS findings remains challenging and requires other biological resources. Sectors of impact. In recent years, Omic sciences have become an essential tool to promote the innovation of different sectors, such as the food industry, environment, or health and biotechnology, in which products or substances of biological origin play an important role. Closing perspectives. Omics, including genomics, transcriptomics, proteomics, metabolomics, and epigenomics, are increasingly used in a broad range of research fields. Integrative analyses applying genome-wide association studies with these omics data are expected to expand our knowledge of complex traits and provide insight into the pathogenesis of complex diseases and their causative factors. Thank you for watching this video. If you liked this video, please smash the like button and subscribe to this channel for future updates.